Let's explore the invisible life. Welcome back to MicroMind. Today, we're breaking down one of the most important bacterial classifications, gram-positive versus gram-negative. Gram staining is a laboratory method that uses a violet dye to separate bacteria based on their cell wall. This helps identify and treat infections faster. Gram-positive bacteria retain the violet stain and appear purple. They have a thick cell wall but no outer membrane, which makes them more sensitive to antibiotics. Gram-negative bacteria don't hold the violet stain. They stain red or pink because of their thin cell wall and protective outer membrane, making them tougher to treat. This table shows the key differences you need to remember for exams and interviews. Examples help memory. Staphylococcus and Streptococcus are gram-positive. E. coli and Salmonella are gram-negative. Use these visual cues to remember. Gram-positives are strong and stain purple. Gram-negatives are slimy and dangerous. Pause the video and answer. Which type has a thick peptidoglycan wall? And which type releases endotoxins? Look at the answers. Which one has a thick peptidoglycan wall? Answer. Gram-positive bacteria. Gram-positive bacteria have a thick peptidoglycan layer, which retains the crystal violet stain, making them appear purple under the microscope. Which releases endotoxins? Answer. Gram-negative bacteria. Gram-negative bacteria have an outer membrane that contains lipopolysaccharides, LPS. These function as endotoxins, which are released when the bacteria die and break down. Crack the core with concept-based MCQs. Question number 1. In a microbiology lab, a student observes two slides under the microscope. One slide shows purple-stained bacteria, and another shows pink. The instructor says, this stain tells us if the bacteria have thick or thin cell walls. What technique is being used in this lab? A. Zeil-Nielsen stain. B. Gram staining. C. Simple staining. D. Acid-fast staining. Answer. B. Gram staining. Question 2. A doctor prescribes penicillin to a patient with a gram-positive bacterial infection. She explains that the bacteria lack an outer membrane, making them more responsive to certain antibiotics. Why does penicillin work well in this case? A. Gram-negative bacteria produce enzymes. B. The bacteria are encapsulated. C. Thick cell walls are resistant. D. There is no protective outer membrane. And the answer is D. There is no protective outer membrane. Question 3. A scientist studying Salmonella typhi finds it resistant to multiple antibiotics. Under staining, the bacteria appear pink, indicating a protective outer layer. What group does Salmonella typhi belong to? A. Gram-positive bacteria. B. Gram-variable bacteria. C. Gram-negative bacteria. D. Mycobacteria. Answer is C. Gram-negative bacteria. Question 4. During an outbreak, two bacterial strains are isolated. Strain A releases exotoxins and stains purple. Strain B produces endotoxins and resists antibiotics. Which feature is true for strain B? A. Gram-positive with thick wall. B. Lacks peptidoglycan. C. Gram-negative with outer membrane. D. Acid-fast with mycolic acid. Answer. C. Gram-negative with outer membrane. Question 5. While reviewing lab results, a student notes that gram-positive bacteria have a single membrane, while gram-negative bacteria have two, making one more resistant. Which statement explains why gram-negative bacteria are more antibiotic resistant? A. Their cell wall is thick. B. They have a capsule. C. They produce more toxins. D. They have an additional outer membrane. Checkmark answer. D. They have an additional outer membrane. Answers of previous MCQs. 1. A teacher in a rural school begins her biology class by asking students why bread grows mold when left open. She explains that such changes are caused by microscopic organisms called fungi. Later, she teaches that other microbes live in our gut, help in digestion, and even produce vitamins. What field of science is the teacher introducing? Answer. B. Microbiology. 2. At a wastewater treatment plant, engineers study the breakdown of waste using bacteria that convert organic matter into safer substances. They emphasize the role of microorganisms in cleaning the environment. Which microbial role is demonstrated in this scenario? Answer: C. Environmental cleanup. 3. During a lecture, the professor states, some microbes are pathogenic, but others are essential. Without gut flora, you couldn't digest fiber or make vitamin K. Students are surprised by the positive role of microbes. Which term best describes the beneficial microbes in our gut? Answer: C. Gut flora. 4. While visiting a hospital lab, Asha observes technicians using a microscope to identify bacteria in a sample. The lab technician explains, we work with microbes too small to see with our eyes, so microscopy is our best friend. Which characteristic of microbes does this passage highlight? Answer: C. Their invisibility to the naked eye. 5. Pharmaceutical companies often culture specific fungi and bacteria to produce antibiotics. Scientists carefully isolate microbes in sterile labs to prevent contamination. Which of the following is a key application of microbiology shown in this passage? Answer: C. Antibiotic production. Thanks for watching. In the next video, we'll look at bacterial shapes and arrangements.